An amazing study on human versus mouse microglia was published in July 2017 by researchers in the labs of Bart Eggen, University of Groningen in the Netherlands, and Sully Marie, University of São Paulo in Brazil, offer a note of caution to mouse modelers. Analyzing global gene expression in human microglia isolated from 39 post-mortem human brain samples, they find that human and mice microglia indeed have a lot in common. Not everything, though. The way microglia age is distinct. Importantly for studies of age-related diseases, the changes in gene expression that distinguish cells from young and old donors show barely any overlap between the two species. The work appeared July 3, 2017 in Nature Neuroscience. One main conclusion is that microglia may slow down with age, as they downregulate genes involved in cytoskeletal dynamics. Many feel this is an extremely important study. More studies will need to be done to determine whether human microglia are really different from mouse, or if they're changed due to their different environment, but clearly this is a start to what we should be doing now, that is, studying real human microglia. The study provides a valuable resource across the disciplines of neuroscience and immunology. The interspecies age-related divergence in microglial transcriptional signatures adds to accumulating evidence that supports the use of human post-mortem and iPSC platforms to complement, but not replace, existing in vivo models of aging and neurodegeneration. For the growing number of investigators who are already switching from mouse to human microglia in their studies, the results provide a needed benchmark, as this is going to be heavily used by a lot of researchers, not only to determine the baseline expression of genes that should be looked for in any human microglia cell culture experiments, but also as a great comparative resource for people to purify microglia from post-mortem brain with neurodegenerative disease, infection, or trauma. The differences with aging are interesting. These genes could become key targets for further investigation of neurodegenerative disease. How did they do this study? Authors Thijs Galitro and Inge Holtman turned to autopsy samples collected either in Groningen or São Paulo. Using fluorescence-activated cell sorting, they rapidly isolated viable CD11b+, CD45+ microglia from parietal cortex samples from 39 people who had died between the ages of 34 and 102 without a diagnosed brain disease. After deep sequencing the transcripts in each sample, the researchers identified a set of 1,297 genes that were expressed at least 8 times higher in microglia compared to an unsorted cortical tissue sample. The genes, which the scientists called the human microglia core signature, fell into expected functional classes, with many related to the scavenger and innate immune activity of microglia. A proteomic analysis, Western blotting, and immunostaining supported the RNA expression data. Eggen's group documented extensive overlap of expression profiles between human and mouse microglia, both from mouse data generated in-house and taken from previously published reports. The results should reassure researchers that human and mouse microglia are extremely similar. They share the majority of genes, they have the same function. They are the same cell type. Nonetheless, differences stood out. Compared with mouse, human cells expressed higher levels of genes related to immune function. Part of the difference lay in human-specific genes such as SIGLECs and MHCs. Part of it lay in upregulation of shared genes, indicating a more active ongoing inflammatory response in humans than mice. With regard to aging, Eggen and colleagues tallied 212 genes that increased and 360 that decreased their expression with age. Hierarchical clustering of the top 100 most affected genes clearly distinguished young and old profiles. But compared with profiles of aging mouse microglia, the aging human microglia were starkly different. Only 14 genes increased and only 9 decreased in both species. The results suggest that human microglia age differently than mice, and do so in an interesting way.